Okay, so thanks for turning out this morning. I know it's going to be a long day, and hopefully this will be really helpful for getting your studying a little bit more, or your last bit of studying if you have time, hopefully focused on some of the aspects of the course, those that I regard as the most important, and they're reflected by that in the exam. The exam's going to have 33 multiple choice questions in it, just like that is the one today, comparable to the ones you had in the last couple sections of the course. And then, as I mentioned, there'll be a set of questions on the final, too, that fewer questions that will reflect mostly the material in the later part of the course as regards structure and function of plants. So let's just get right on with it here. I've basically gone through and pulled slides from the lectures that hit the main points that I want to emphasize. And so you remember, of course, that we covered a really broad array of groups across the tree of life in this part of the course. And for reasons that became obvious during the course, that photosynthesis evolved, or actually was acquired multiple times across 4 of the 5 major clades of eukaryotes. But we started out with fungi, which are actually more closely related to animals than to any of the photosynthetic lineages, for reasons that they were traditionally included in botany. They have cell walls, they have largely immobile, they're largely immobile during most of their life cycle. And then we moved on to all the photosynthetic organisms, which include the various groups of algae and the land plants. And in general, these major groups, I had a few things to say about their biology, their ecology, and those major characteristics are good to review. And we'll be looking at those as we go along here. But I'm not going to talk about all of these. But for example, for the large, the aspects like whether or not organisms are multicellular or unicellular, in the case of different kinds of algae, what their ecology is as far as the kinds of habitats that they be found in, just very general things. Hope that you would pick up on with regard to these various major groups. The life cycles also really want to focus on the big picture first. We have 3 major types of sexual life cycles in organisms. We mostly dealt with the plant life cycle, the land plant life cycle that is an alternation of generations. But you should also be able to compare that to the animal life cycle and the kind of life cycle we saw in fungi with regard to the relative timing of meiosis and fertilization and the consequences of those, the presence or absence of mitosis at the two at diploid versus haploid stages as regards consequences for the types of organisms that we get in these life cycles. So in animals, remember, we just have a diploid multicellular organism. Fertilization occurs right after meiosis. We just have gametes that are haploid. And in fungi, fertilization is followed immediately by meiosis, so our zygote's the only diploid stage. We only really have a haploid organism with the insertion of that dikaryotic phase, which is just basically where fertilization is the 2 aspects of it are separated in time, and we're going to go over that again in a moment, plasmogamy and karyogamy. Yeah, I guess something's wrong with this system here again. They've had some trouble with let me see if I get the volume up here. Is that better? Okay, great. Thanks for mentioning that. So then the fungi, we should review the general characteristics of fungi that set them apart and that make them similar to other botanical organisms. Remember, they have unusual cell wall composition, but they do have cell walls like plants. Chitinous cell walls, though. They don't store their energy in a way similar to other plants. It's not stored as starch, but as glycogen like animals. And there are a few things like that that are interesting about where there's convergent stored plants, but similarities with animals. Just a few characteristics there on an absorptive type of nutrition. And then we mentioned these 5 major phyla of fungi. But we really only went into great detail about the life cycle and biology of the Basidium mycota, the higher mushroom group. But we did talk about how these other groups that branch off early in the tree don't have the septate hyphae, they have the open hyphae. They don't have fruiting bodies that are very large, at least. And also mentioned that these groups aren't all the ones that have been historically called fungi. Remember the mycota, the water molds that have hyphae and look fungi-like are actually more closely related to some of the algae in that straminopyle group that includes brown algae and diatoms. And the slime molds we just mentioned briefly as a group that is somewhat animal-like and somewhat fungi-like. And it's actually is in the same major clade that includes fungi and animals, but is an earlier branch, so it's no more closely related to fungi than animals. And then the fungal life cycle, remember, as we just mentioned, you have meiosis immediately following the last stage of fertilization, so you only really have a haploid phase germinating from the spores, but then we have an extended fertilization period with the fusion of the cytoplasm of the hyphae long before the fusion of the nuclei that results in this dikaryotic stage. Hello? Okay. Well, the dikaryotic stage is the one that the mycelium basically is composed of throughout the majority of its life in the Basidium mycota. So that's yeah, okay. Interesting commentary there. So also remember this group Mycota, which is kind of a taxonomic garbage can for asexual groups of fungi. And the main reason I mention it is just because so many of the fungi are strictly asexual, they have been hard to classify, and ecologists still find this classification useful since asexual fungi have some features in common that warrant having a group recognized to include those. But most of the members of this group have been moved off into other phyla of fungi as their relationships have become clear from molecular phylogenetics. But this, if you don't know the relationships of an asexual group, this would be the first place you would insert it in the classification system before it got sorted out molecularly. Also, we talked about these symbioses involving photosynthetic organisms and fungi that are not only really important for fungal diversity, but probably responsible for the success of land plants and their ability to colonize land originally, most of them being associated with either an arbuscular mycorrhizal fungus or ectomycorrhizal fungus. This being really, these associations often being either critical for establishment of seedlings, or else they do help the fitness of the plant that has this type of association. So these associations are really critical for ecology and terrestrial ecosystems. And we talk about lichens as well, which involve a unicellular photosynthetic organism, either a green alga or a blue-green bacteria, a cyanobacterium. All right, so remember the algae are polyphyletic and widely scattered across the tree of life, and that's why we consider them in quotes here. And remember the reason why that we have photosynthesis scattered all over the tree of life is because of these capture events that resulted in not a convergent evolution of photosynthesis, oxygen-generating photosynthesis, but co-opting of it by these different eukaryotic clades that either started out by capturing a cyanobacterium in an early heterotrophic eukaryote and engulfing it and having it fully incorporated into their cell as a plastid, a scleroplast, or there was a secondary endosymbiotic event where the green alga, a unicellular green alga, or unicellular red alga was captured, and those were then co-opted as chloroplasts with the nuclei becoming vestigial inside those chloroplasts or disappearing altogether through time. So the red algae, remember, and the green algae, as well as the land plants, which descend from within the green algae, descend from one of these from the only primary endosymbiotic event that we definitely can hypothesize with certainty. And then we have either capturing of the green and red later to give rise to all these other groups of photosynthetic organisms. So that's how we end up with photosynthesis scattered across 4 of the 5 major clades of eukaryotes. One of those clades of eukaryotes is the one that includes greens, reds, and land plants from the primary event, and the other 3 are these 3 here, which are widely scattered across that eukaryotic tree. That's a little more detailed than than is really necessary here, but just to make the point that this endosymbiotic capture is what led to all that photosynthesis across the tree of life. And also, it's important to keep in mind the evidence that supports the endosymbiotic theory as it pertains to the evolution of photosynthesis in different groups of eukaryotes. And remember, I went through the evidence in a series of slides, the size of plastids and mitochondria relative to the size of prokaryotic organisms, the way they replicate by binary fission like a prokaryote, the size of their ribosomes inside these plastids and mitochondria that's similar to the ribosomes of prokaryotes rather than the ribosomes out in the cytoplasm of the eukaryotic cell, antibiotic susceptibility similar to some bacteria, and the genomes are circular inside both chloroplasts and mitochondria like those of prokaryotes. So there's pretty good evidence there for the endosymbiotic theory as it pertains to this particular aspect of biology. And then remember we talked about the origin of land plants being derived from within the green algae. They descended from within the green algae, most closely related to a group of freshwater green algae. So not marine species, not out in the ocean, but in the freshwater. So we can see evidence here for sort of stepwise progression to land environments from saltwater to freshwater on land and then to land itself. These freshwater green algae sometimes being multicellular and having only a haploid phase, the ones that are most closely related to land plants. There's no alternation of generations in the closest relatives of land plants. I mentioned an example of alternation of generations in the sea lettuce, but that's a very distant relative that's evolved alternation of generations independently of land plants. And we have basically the insertion of the sporophyte generation into the life cycle of the land plants and the common ancestor of the land plants. And so not surprisingly, in these early branching lineages, the bryophytes here that are paraphyletic grade at the base of the vascular plants, so all these are vascular plants, here are the vasculars, I'm sorry, all these are land plants, and here are the vascular plants. We have gametophyte dominant in the 3 major lineages of bryophytes, which reflects this origin from algae that only had a haploid phase, where the only diploid stage was the zygote. Here we have the mitoses after formation of the zygote, but we still have retention of the sporophyte on the gametophyte and dependency of the sporophyte on the gametophyte. And so one of the big trends in land plants is the progressive dominance of the sporophyte generation and the potential there for more complex gene expression and the potential for more complex organisms evolving that we see when we get into the vascular plants. But in all these life cycles in the land plants, the alternation of generations, we have multicellular diploid generation, the sporophyte, multicellular haploid generation, the gametophyte, and the sporophytes produce sporangia. Sporangia is part of the sporophyte, wherein meiosis occurs to give rise to spores, the spore-bearing plant. We have mitoses, of course, giving us this multicellular diploid organism after fertilization. We also have mitosis in the germination of the spores to produce the haploid organism, the gametophyte. And gametangia are produced by the gametophyte. So remember, gametangia are part of the gametophyte, and they produce gametes by mitosis in the case of alternation of generations. Okay, so the moss life cycle we showed, and this is again the one with like the other bryophytes, the gametophyte's dominant, the sporophyte is attached and independent and relatively short-lived. One type of spore produced in bryophytes from one type of sporangium on the sporophyte. And in this case, though, even though we have only one type of spore in terms of its size and the type of sporangium it's produced from, we can get unisexual gametophytes. Male gametophyte, photosynthetic, as is the female, that has gametangia again. Remember, there are 2 types of gametangia. Antheridia produces sperm, archegonium produces eggs, and fertilization occurs in the archegonium. And the sperm has to swim to the archegonium, so freestanding water is required for the life cycle to be complete. And we have an embryo stage for the first time in the land plants. So embryos are a novelty of land plants. This can be a resistant or resting structure, or resting stage that we see more importantly later in seed plants. But it's found throughout the land plants, and it's part of the reason why we call all the land plants the embryophytes. We didn't introduce that term, but that's the technical name, the taxonomic name for the land plants, the embryophytes. And then the sporophyte is developing directly out of the gametophyte. So the fern life cycle, the one that we presented at least, is similar to the bryophyte life cycle in the sense that we again have a free-living gametophyte generation, and so it's exposed to the environment. It's either photosynthetic like shown here, or it has to live in association with a fungus underground in some species, but it's not dependent on the sporophyte. It's living by itself. It's like the mosses that we looked at. We have one type of sporangium, one type of spore. In this case, one type of gametophyte. It's bisexual. It produces both archegonia and antheridia on the same individual plant. And again, freestanding water is required for fertilization, so it has some of these same features of bryophytes that are retained from a common ancestral organism that was ancestor to all land plants, where we have requirement for freestanding water. Spores are the dispersal unit, as in the mosses and in the bryophytes. But these are vascular plants. The sporophyte's dominant. And as the sporophytes briefly, we have an embryo stage still, as in all land plants. The sporophyte germinates within the archegonium, and it emerges from the archegonium, as in mosses, but then it becomes free-living, uses the gametophyte dies, and we have this, what can be a pretty large sporophyte, long-lived sporophyte with an underground horizontal stem or rhizome, usually the only stem that we get in ferns. And the sporangia are born on these structures called sori on the underside of the leaves that you can see with your naked eye. The sporangia are too small to see with your naked eye in any detail, but the sori stand out as these often circular patches on the underside of leaves where we have many sporangia clustered, and that's from where we have release of spores. Okay, so talk about some of the early adaptations to the land environment. Desiccation-resistant spores, a cuticle, stomata. This cuticle being poorly developed in bryophytes compared to what we see in the vascular plants, but on the sporophytes of mosses and hornworts, we do have a pretty well-developed cuticle as well as stomata present that allow for gas exchange. But in the gametophytes of the mosses, remember, we have desiccation tolerance, and so these things aren't particularly good at preventing desiccation, but they live through it, unlike most vascular plants, except when they're in the seed stage. Gametangia are a novelty of the land plants, protecting the gametes inside of a jacket of cells. Embryo stage we already talked about, being found throughout the land plants. The fungal association we already talked about as being important in capturing, absorbing nutrients and water from the soil. And a rich secondary chemistry, this defensive chemistry we talked about right at the end of the last lecture, also important for UV protection. And vascular plant innovations, the dominant sporophyte again, well-developed cuticle and good ability to resist desiccation in the land environment. Vascular tissue allowing for efficient conduction of water and nutrients throughout the plant. This vascular tissue, including xylem that's lignified, that is tracheids and vessel elements, lignin imparting strength and structural support against gravity, allowing this to become a large branch sporophyte, which we don't see in the bryophytes. And roots evolving in the vascular plants, allowing for absorption from deep water sources and anchorage of a big sporophyte. Heterosperity, remember, you should also remember heterosperity is a concept, a condition that we see. It occurs in lycophytes and ferns to a very limited extent, but we didn't really talk about it other than to show you this one slide of a lycophyte that has heterosperity, where you see the large megaspores and small microspores really clearly. But this is the common condition of all seed plants. All seed plants are heterosporous. They have heterospory, and that allows for the evolution of the ovule, which matures to be the seed. We have the female gametophyte completely enclosed within the megasporangium throughout its life in the case of the seed plants. But in all heterosporous plants, the gametophytes develop inside the walls of the spore. So we have very small gametophytes with heterosperity, and they develop inside the spore wall. The spore wall expands a bit in seed plants to take up the whole megasporangium in the case of the megaspore. But this is protection of the gametophyte generation, reduction of the gametophyte generation, sequestering of the gametophytes, and that's taken to the extreme then in the seed plants, where we have the sequestering of the female gametophyte. It never leaves the megasporangium. The megasporangium is encased in integument. The immature ovule is, remember, in a megasporangium with integument around it. So that's the high level of protection of the gametophyte generation, which is nourished completely by the sporophyte that the ovule is attached to. And so remember that we only get one megaspore that actually is functional inside the megasporangium, which again in all seed plants is encased inside integument, which together makes up the immature ovule, which ultimately becomes a seed, but not until we have the female gametophyte germinating, developing. We have fertilization of its egg, and then that egg develops into an embryo, and then finally we have mature seed with the embryo going dormant, desiccating, and the integument hardening to make a seed coat, and the seed being dispersed away from the parent plant. So then we don't have free sporing condition. Remember, spore is just a unicellular dispersal unit. There's not much nutrition there for establishment of the new plant, but in this case we have all this nutrition inside the seed. The embryo is already partly developed. It has a really big resource it can draw on as it's getting established, the new sporophyte's getting established. Remember, the pollen is the microgametophyte, the male gametophyte. Immature male gametophyte's the pollen grain. The pollen is dispersed out of the microsporangium, unlike the female gametophyte, which never leaves the megasporangium. And it arrives at some sort of receptive surface in all seed plants. In gymnosperms, it arrives at the ovule itself. We have the pollen tube grow down, and a mature male gametophyte is then the pollen tube, okay, that grows down to the egg and fertilizes it. So again, we have all this nutritive tissue for germination of the embryo in the case of seed plants. We've gotten away completely from the need for freestanding water, so this is another adaptation to the land environment. There's no free-swimming sperm stage out in the open environment. Again, the male and female gametophytes of all seed plants, including the pine, are sheltered from the environment. Even the pollen grain, when it's transported by some intermediary agent, has a protective pollen wall around it. So there's quite a lot of sheltering here. Also, of course, the seed coat's going to protect the embryo inside the seed, and we have all this nourishment, both of the female gametophyte inside the ovule as well as the embryo as it's developing, and even after it's dispersed as it's getting established as a seedling. But in angiosperms, we have a number of innovations that are different than in the pines because the pollen's delivered to a stigmatic surface, not to the ovule itself. So we have this selectivity on the part of the sporophyte parent of the female gametophytes inside these ovules that's going to either prevent germination of self-pollen or kill the pollen tubes that are growing down in there if it's a self-incompatibility reaction, if this pollen is genetically too similar to the sporophyte here. And also we have male-male competition, these pollen tubes competing with each other for ovules as they grow down. And we also, of course, the carpel is what's the main innovation here in the flower that allows for all this.